Hello students. So we have learned about pollination, and we know that pollination is a process where we are bringing together the male and the female gametes. And in this process, a compatible pollen grain, if it is going to come and sit on the stigma, it will lead to the post-pollination processes, right? For example, the pollen tube growth and the release of the male gametes leading up to the fertilization. But today in this video we will talk about another process called controlled pollination, right? So controlled pollination, as the name suggests, we are going to regulate the pollination process. Regulation of pollination process, how? So we are going to regulate the type of pollen grain, right? So what type of pollen grain is going to pollinate a particular flower, that is going to be regulated here, and thereby controlled pollination. So why it is important and where it is important? So this particular process is important in crop improvement programs. And why is this process important? Because in this process of controlled pollination, we can achieve something like combining the traits or combining the superior traits of two different species or even genera and produce a commercially superior, right? It is going to be more superior than both the parents, commercially superior varieties, right? So this is a place, crop improvement program. So in the crop improvement program, we use the controlled pollination to get something called commercially superior varieties, that is going to have the traits of two different plant species here. As I said, we use the type of pollen is going to be regulated, that is we're going to use desirable pollen grain only. Of all the pollen grains that are used, the desirable pollen grain that is used, right, for pollination process. And we do another thing, right? One is going to be using the desirable pollen grain for pollination process, and second important fact we have to keep in mind is the stigma, right? So protecting the stigma from unwanted pollen grains, right? And that is called the preventing contamination, right? When we say unwanted pollen grains, we say this as pollen contamination. So we will prevent this also, pollen contamination. So what is the technique involved here? Two technique. First is called emasculation and second is called bagging, right? We'll talk about that. So when we talk about emasculation, emasculation refers to the removal of anthers, right? So when we are having the flower in a bud stage, right, what we do, we are going to remove the anther before the dehiscence, right? It is important to remove it before dehiscence because after dehiscence anyway the pollens are released and the pollen grains are going to anyway sit on the stigma, which is going to not allow us to regulate the desired pollen grains for pollination. So the same flowers pollen grains are going to pollinate itself. And the second technique, that is the emasculation, removal of anther, the second technique is called bagging. So as we are talking about the pollen contamination, right, undesirable or unwanted pollen grains, we need to prevent that. And to do that, what we do is we actually bag means we cover the entire flower after emasculation or emasculated flower in a bag, and this bag is typically made up of butter paper, right? So butter paper bag. Another thing that you have to keep in mind that I forgot to tell is in emasculation what we do, we can use a forceps, or we can use even the scissors, or there are other methods also, but again using the forceps is one of the easier technique to achieve the emasculation process. Now the question is, how are we going to bring about pollinations? So imagine that you are going to have the emasculated flower, and once the flower is emasculated, the stigma is going to mature. So once we have the emasculated flower, so we will wait till the stigma is matured. Matured stigma is formed, and once we have the matured stigma, we will bring about the matured pollen grain, right? From where we will get, so whichever plant is having the superior trait, from that plant's flower, actually we are going to bring the matured pollen grain, and this matured pollen grain we are going to dust it, we are going to place it on the matured stigma of the emasculated flower. Now once you have the matured pollen grain having pollinated the matured stigma of the emasculated flower, we actually go for, once we do this, we can go for a process called rebagging. Rebagging, why? Because again, after the matured stigma has been given the right pollen grain, we do not want any sort of pollen contamination to ensure that they are being pollinated by the desired pollen grain. So this is in short the process of controlled pollination. And after the rebagging, what we do, we allow the post fertilization process. So we allow further development, and when the process continues, it will give us a progeny, it will give us the outcome that is going to be resulting from these two desired superior parents only, from where we have taken the pollen grain and the flower in which we have performed the emasculation. So remember, this is the case in a typical bisexual flower. So now the question is, what happens in a unisexual flower? So let us talk about the process quickly here, and then we'll continue to what happens in unisexual flower. So as you can see here, we are going to have the bisexual flower here with the stigma and the stamens, that is representing the female and the male components in the flower. And here we are seeing the process of emasculation, where actually they are removing the anther. As I said, the easiest method is to use the forceps or scissors and cut out the anthers or the stamens in the early stage or the bud stage. And once we do that, we are going to go for bagging, and if the stigma are matured, we are going to allow the pollen transfer from the desired plant. So here we are going to have the plant from which we are going to take the pollen grain. There is some superior quality here and there is some superior quality here. So this is our emasculated flower that is going to represent the female here, right? Because we are going to remove the male component. So it is going to act as a female flower, and this is going to act as a male flower. For this process, again, both of these are, remember, bisexual flowers. So once we transfer the pollen grain from the desired source, right, we are going to dust it on top of the stigma and allow the further process to continue. So when this flower is going to develop, that will give us the progeny, right? And this progeny is going to be the superior progeny that we are talking about, commercially superior progeny that we are going to talk about. Now the question is, what is going to happen in case of the unisexual flowers? So let us talk about that. So in case of unisexual flower, we know that the flowers can contain either the male or the female component in a flower, right? There is no case that we're going to get both the male and female component. That is your carpel and stamens together in the same flower. The flower is going to contain either the anther and filament or the stigma, style, or ovary. So here there is one thing that you have to keep in mind is no need for the first step, that is no need for emasculation. That is you have to keep in mind, because there is a separate female flower available. And here what we do, we use the female flower, we use a female flower bud, and we are going to bag it, right? Bagging process before the flower is going to open. So the female flower bud, we are going to perform the bagging before the flower opens, right? So once the flower is bagged, there is no chance, right? So why it is important? We can say that it is going to prevent pollen contamination, right? And next what we do, once the stigma here in the female flower, right, once the stigma matures, desired pollen grains is dusted on it to bring about pollination. Right? So this is what we need to understand in unisexual flowers, that there is no need of emasculation because we have a separate female flower. And what we do, we start bagging it before the flower opens, and that will allow us to prevent any sort of contamination by any unwanted pollen grain. And the moment the female flower here actually getting maturation, that is the stigma is going to mature and it is receptive enough to the pollen grains, we will take the desired pollen grain from the desired flower having the desired character or traits, and we will dust it on the stigma of this bagged flower, and that will bring about pollination, thereby the controlled pollination. So dear students, this is what you have to understand with the controlled pollination process, that is going to help us in a process called, remember we are talking about artificial hybridization.